Well, good morning to all of you, and this is Dr. K.V. Shashidhara. I'm a senior scientist in Medicinal Chemistry Division of this institute, Central Drug Research Institute. We are having a brainstorming session as a part of this workshop, which is entitled Renewing the Tradition of Natural Product Research in India. As part of that, we have stalwarts over here. This is Dr. Goverdhan Mehta, as you all know. He's a National Research Professor, School of Chemistry, University of Hyderabad. He's an eminent scientist, as you all know. And previously, he was former Director of Indian Institute of Science, Bangalore, and Vice Chancellor of University of Hyderabad. His total area of research, if we say, is the synthesis of complex natural products. On the other side, we have Kupuswamy Nagarajan. He is an eminent natural product chemist and medicinal chemist. He is currently, presently a corporate advisor to Alchem and several companies, including a consultant to several companies. And he is also the RC chairman of RC member of CDRI. And this time we have Dr. Madhu Dixit, director of CDRI, an eminent scientist. And towards the end we have Dr. T.K. Chandrasekhar, former secretary of SERB. So this is a kind of brainstorming session. We are trying to address some challenges that are currently facing the natural product research in India. So let me start by asking Dr. Goverdhan Mehta that in spite of a lot of success in natural products, if we say that 40 to 50% of the drugs that are on market are plant-based or plant-derived, in spite of that, the natural products have seen a decline. The pharma companies are not in favor of putting up this. What are the bottlenecks? Is it the supply issues or IPR? What are your comments on that?
Natural product research is traditionally time consuming.
Right.
And in some ways, if I can say, it is cumbersome. You have to have collection, isolation, dereplication, and then biological evaluation. Many of the pharma companies are structured basically to deal with pure compounds that can be synthetically made, and then you subject it to the regimen which is followed from HTS to biological, clinical trials and so on. So intrinsically, two reasons in my opinion why pharma does not take that much interest, because they want to avoid the cumbersome roadway or roadmap for getting access to these compounds. The second is that they believe that the natural product based drugs eventually, despite their success and so on, may not be amenable to the kind of scale up that pharma industry requires. Because there could be a lot of biodiversity related issues, etc. Okay. Because if you have a blockbuster, it is required in huge quantities.
Right, exactly.
Okay? And there could be lots of complications with regard to collection of material and separation and so on. Now, another reason for this is that the countries, with the exception of India perhaps and China, the countries who have traditionally been very strong in drug discovery are not necessarily rich in biodiversity and access to plant products.
Right.
So they will have to depend on those geographical regions from where they can get, and they don't want to take that kind of a risk. So these are, frankly speaking, main reasons for this. But now that the overall geopolitical dynamics and the economics of the world is changing, it is not necessary that countries like India, China and other emerging countries should follow the same model. Because if we are intrinsically rich in our biodiversity, and India harbors about 8% of Earth's biodiversity, we should rely more and more on our resources. But we should be very careful that you don't endanger a particular species by over-harvesting and so on and so forth.
You have really exemplified the example of halocontin and eribulin, which is a simplified structure. See, when we have a complex natural product, not all parts of the molecule will interact, only a part of the pharmacophore is essential. And what do you think is the simplification of natural product is a viable strategy as far as natural product
Of course, obviously, because we must understand that nature makes natural products not because human beings can use them as drugs. They are products of secondary metabolism, as we know, and that's why they are secondary metabolites. But the important thing is, and that's what I also try to highlight, that they have been forced in a complex biological environment through all kinds of interactions which we cannot today replicate, because this has gone through thousands of years of evolutionary interaction. Okay, so they harbor certain memories which are intact. Now, the challenge therefore for us is that can we remember these or we can unlock these memories and make them relevant for what we want to do. And that's why I even broadened this discussion, and I really throw a challenge, and I think that's where the future is, that do we know how to do a fractionation of biological activity through structural changes. Okay, it's a huge challenge. But today we can use the tools of bioinformatics. We have better understanding of the targets, so you don't have to do randomly. I can tell you that eribulin evolved from halochondrin because it was identified that the binding domain rests in the yellow portion that I showed, and therefore it's a why not chop it.
Then that leads me to another question. When we showed the hashtag structures, there were a whole lot of diverse structures targeting a single target hashtag. And that applies truly when we talk of microtubule stabilizing agents like Taxol, eribulin, apothilone, all diverse class of compounds targeting microtubule. So how do you take that challenge?
It's still in the realm of the unknown. It only means that during the evolutionary process, the binding domains of a molecule, the target, was not necessarily only meant for one possible target. We also know that many of these large molecules also manifest their activity through allosteric effects. So even the target modification to have the efficacy is a very important area. So therefore, I'm not surprised. But what the message from this, and that's why I put it, that from a 3-carbon simple, allicine from garlic to a pentacyclic, hexacyclic complex molecule effect the HDAC inhibition clearly therefore shows that we have wide variety of choices. We should try to understand this. Why is it so? This is unknown story. I mean, I think we need to dive deep into it.